All right, folks. I feel like I need to go check and see if I'm live. It says I'm live. It says people are watching. Yeah. So there we go. And you can hear me. Let's just make sure the right microphone is working. Currently, it seems to be. Yeah. All right. Okay. So we're looking at Unit 4. If you have questions about a specific section, feel free to ask. Otherwise, I am just going to sort of work through the Khan Academy questions like I've done in the past. I'm actually just going to have to excuse myself for a second to go get a drink because I can already feel it. I can already feel the throat thing. So one moment, be right back. Much better. Much better. Okay. So I'm looking at the sections here in Khan. Khan has one more section than what we covered, but this content's going to be the same. Actually, I say that. Looks like their sections are caught up with ours, actually. Well, that's nice. So I'm going to do these quizzes. There's two quizzes and then a unit test, and so that's what we're going to do. The first quiz is 4.123, and the second quiz is 456, and then the unit test, of course, is all of them combined. Haven't looked at these questions ahead of time, so I have no idea what I'm getting into. Here's some epinephrine. Man, that's great that it wants me to reach new levels. I think that's fantastic. I'm going to try to include all the things. This is a long one because it's got that epinephrine pathway that you've seen on an FRQ. All right. So here's our question. So again, I see this question on the test. I'm just orienting myself. I may even orient myself before I go down to look at the question. Not too much though, because you don't know what the question is going to ask you. But I can see signal transduction pathway. I can see epinephrine. And you know what? I think it would be beneficial. I didn't have a lot of you guys come in and talk about the FRQ. And so let me break this picture down for you. I think that would be helpful for some of you. And so I'm just following the arrows, right? I can see that epinephrine is received at the epinephrine receptor, which is on the cell membrane, which is then going to activate adenylyl cyclase, which is going to activate cyclic AMP or CAMP, right? And CAMP's like a secondary messenger here. It's like just a relay molecule. And CAMP is then going to turn on two separate processes, right? And I think some of you guys missed this on the FRQ. One molecule, two processes, right? And one of those processes is active glycogen activate, or to make it active, glycogen phosphorylase. And what is the job of glycogen phosphorylase? It looks like it's taking apart glycogen to make glucose, right? And then CAMP also activates this over here, which is an inactive, or causes glycogen synthase to be inactive, which causes no new glycogen to be made. which makes sense. Glycogen synthase, literally making glycogen, turning it off. And so think about that just for a minute. What's going on here? This is unmaking glycogen here on the left, right? And so we don't want to be making it and unmaking it at the same time. And so essentially what epinephrine is doing, it is causing glycogen to stop being made and to start being pulled apart into glucose, which can be used for more energy. That's what's going on in this picture. Now let's go down here to the question. The image above depicts the signal transduction pathway of epinephrine. During one step in the pathway, the enzyme glycogen synthase is phosphorylated, which inactivates the enzyme. If a toxin prevents the phosphorylation of glycogen synthase during this pathway, which of the following will most likely occur when epinephrine comes into contact with the receptor? So if what will likely occur, the phosphate in this case turns off the glycogen synthase, and so if that prevents that from occurring, then glycogen will be made and being unmade at the same time. So you'd have glucose being made and glycogen being made at the same time. Let's go find that in ABCD. Glycogen's phosphorylase will phosphorylate glycogen synthase to prevent new glycogen from being made. I have no reason to believe that. Again, it's weird. Makes the picture weird. Epinephrine receptor will no longer recognize the no. No need to reason to believe that either. Right? Glycogen breakdown will still occur, but glycogen synthesis will not be affected. C is where I'm going. Glycogen is being broken down and being made at the same time. Just love that little affirmation noise. Here we go. Here's another. And I end up having to zoom in quite a bit. This one seems all formal. The following is an excerpt from a scientific paper written by Levy et al. Cell wall's barrier to virus exit. This plant cell wall is a barrier to virus exit from and entry into the cell. Okay. Thus, successful infection requires that plant viruses encode movement proteins to transport the viral genome locally, cell to cell, within the leaf and on the vascular system, and on into the vascular system, through which the virus will systematically invade the plant. Okay. So basically what this is saying is that viruses can't move through the cell wall, right? And so successful infection for this virus means that it not only is doing things viruses normally do, but is also creating proteins that help move it through so it can get into the vascular system of the plant. Which of the following best predicts how movement proteins help plant viruses travel from one cell to another? They allow passage through the cell wall in some way. Let's go find that. I don't know what this has to do with signal transduction, but we're going to find out. Movement proteins increase the movement of transport vesicles between plant cells. Vesicles between plant cells just doesn't. I'm struggling with that one, but let's wait. Movement proteins increase the movement of vacuoles between plant cells. We know vacuoles aren't moving. This is a weird question. Movement proteins increase the surface area of the no. Movement proteins increase the permeability of the plasmodemata between plant cells. Okay, so this is back to 4.1. We talked about examples of direct contact in cells, and I mentioned a couple of examples of how cells can communicate, and one of those things was plasmodesmata, which are little holes between plant cells, and so that is what makes sense to me. These other three don't even make any sense, right? Because vesicles are staying inside the cell, vacuoles are not moving. C is, literally D. If you don't know what plasmodesmata is when you come to this, D is really the only. Watch me miss it. No. Okay. All right, then. Here's another one. This is another. I love these pictures. I just wish I'd have learned this stuff when I was in high school. College would have been so much easier. It sounded like I was like selling myself there, but it just. The information below shows and describes the intermediate steps of MAPK-ERK signal cascade. Okay, it does. After MEK is activated, it phosphorylates and activates ERK. ERK goes on to phosphorylate and activate a variety of target molecules, including transcription factors like c-MYC, which promote growth and division. All right, so we see that. We see that MEK is turning on ERK, and ERK is turning on c-MYC, which is causing the transcription of a gene, which is essentially gene expression. So let's switch the following. Toxins will most likely inhibit phosphorylation of c-MYC. Okay, so anything that's going to turn off any of the above processes is going to prevent that. A toxin that prevents the phosphorylation of ERK. Okay, that's it. I don't even need to look at the other one. Because when ERK's phosphorylated, then that. So I'm not even looking at the other ones. I'm going to stick to my guns here. Yep. Because if you prevent that, the whole thing's off. And so I don't even know what those other ones say. It's not important. All right. Ooh, this one's, man, let's see how long we can get a question to be. This one's intense. It's like a third of a book. It's got a big graph on it. Let's see if I can squirm it in between here. Oh yeah. It was almost too big. So the letters are a little fuzzy. To complete external fertilization in fish, sperm cells must swim through open water in order to fertilize fish eggs. During this process, females release a substance called ovarian fluid, which helps guide sperm cells through egg cells or toward the egg cells. Researchers hypothesize the sperm cells are mainly attracted to the ovarian fluid released from the same species. To test this hypothesis, researchers performed an experiment in which they studied the effects of salmon ovarian fluid and trout ovarian fluid on salmon sperm. In each trial within the experiment, the researchers placed the salmon sperm on one side of the porous membrane and either salmon or ovarian fluid or water on the other side. After 2 minutes, the researchers counted how many salmon sperm passed through the membrane and fluid. The results were printed as below. The salmon sperm went more to the salmon fluid. The trout sperm went more to the trout fluid, which is exactly what we'd expect to see, right? And notice too that the opposite species is as much as water, right? Even though it looks like it's more, the bars overlap, and so statistically, the opposite species is no more than water. And so what this is saying is like it's just random between these two, but this is significantly different. Same over here. Which of the following conclusions best supports the above data? And so we're just talked about the data. Let's go see if anything matches up with what we said. Salmon sperm respond more strongly to local regulators in the trout ovarian fluid. Nope, they don't. Salmon sperm respond more strongly to local regulators in the salmon. Yeah, B. Okay, so the difference here is local versus long distance. Remember, local is like around a group of cells, right? As opposed to long distance, which would be like bloodstream sort of thing. What? Maybe it is long distance. Did I not read it correctly? I guess that, I don't know. Yeah, that's a weird question because unless I just missed something that denoted that it was distance. Maybe the open water word there. Yeah, no, it's just a bad question. All right, it's weird because it just didn't define those words in the context. So we didn't like the salmon question. It's a good question until that part. All right, let's look. Following is an excerpt from the scientific paper by Jean-Pierre Rothman. Cholera, an infectious disease that can kill humans within a matter of hours, is a manifestation of a highly efficient and specific toxin delivery mechanism. This mechanism, which is shared by several bacteria, delivers a potent toxin to activate selectively the adenylyl cyclase system of human intestinal secretory cells. This increases production of the secondary messenger cyclic adenosine monophosphate, or CAMP, that activates an intestinal crypt cell chloride ion channel, thereby resulting in massive fluid and electrolyte secretion into the lumen of the gut. Interesting. I knew what cholera was, but I didn't know how it worked. Now I do. Based on the information above, which the following is most likely to be observed in the intestinal environment of a person infected with cholera. So let's look. This toxin, potent toxin to activate the adenylyl cyclase, which is sort of a membrane enzyme that then activates cyclic AMP, which is a secondary messenger that activates an intestinal crypt cell chloride ion channel, thereby result. And so what happens is it looks like this causes chlorine ions to go into the intestinal lumen, which then causes water to flood into the lumen from the cells, which is like osmosis, right? Based on the information which the following most likely observed in the intestinal environment, elevated levels of extracellular chloride is what I'm going to go with here, just like I was saying. Because if it puts the chloride out into the intestinal lumen, then it's going to cause water to rush out of the cells and flood the lumen with water. Yep. Just can't have a universal definition, I'm assuming. What she meant. So yeah, it does. Like it's trying to find. I've just found like random picture with it, so you can kind of see. It is so cyclic AMP is always a secondary messenger. So here's just, I have no idea what this is, right? There's a lot going on here. Right? Who knows what is going on here? But notice what's going on here in the middle. It is cyclic AMP, right? And so this is something that is not that toxin that we just talked about. This is something that is not epinephrine, right? But cyclic AMP is activated by several pathways. And so you can think of it as like a generic relay molecule. It is not specific, it does several things. So I think I don't know if that answers your question or not, but. was surprisingly loud. Made me fall out of my chair. Made an 80 on that one. Should have to do better next time. All right, then we're not going to take that quiz again. All right, so here's quiz 2. This one's got 5 questions, supposedly 5 to 10 minutes. It's going to take us a little longer than that. So this one is 4.4, which is feedback and cell cycle stuff. Genetic mutation causes somatic cell to become cancerous, which the following best is the most likely explanation for how the mutation caused malignant transformation. So we know that cancer is any disruption to the cell cycle. So we're going down to find something in that realm. Mutation created an underactive version of a protein that promotes the cell division. So now that'd be the opposite of what we're looking for, right? Mutation created an underactive version of a protein that promotes apoptosis. That could be it. Let's keep going. Mutation created an overactive version of a protein that prevents. No, because overactive prevention would be more prevention, right? Mutation created an overactive version of a protein that inhibits. Again, that's the same thing as C, or yeah, same thing as C. And so we're going with B, the correct answer. We knew that on B, but every once in a while I like to just, get a little wild, read the whole question. All right. Oh wow, literally. It's just this is really funny. Okay, it's like the question is one of the pictures didn't load or something, and so it's kind of dumb, but I think I can read what's going on here. Why don't you just go right back up in there? Okay. Here we go. Image below depicts a somatic cell before DNA replication. Somatic cell is just a fancy word for any sort of cell that's not a sex cell. Yes, I will go over negative and positive feedback. Thank you for that question. For sure. I'll go over that after I finish this quiz, because there'll probably be some of those questions on this quiz. If this cell undergoes mitosis, which the following correctly depicts the daughter nuclei that will result. Okay, so let's look at this again. This is important. The real important part here is that the cell that we see above is before DNA replication. So we know that cell is in G1 phase, which is the normal life of the cell. That cell has 4 chromosomes. After mitosis, we'd expect to see 2 cells with 4 chromosomes each, right? And A was not kind enough to give us a picture, but it says 2 daughter cells, each containing 4 chromosomes. That is the answer, even though we don't get a picture. It was pretty simple. Here's a feedback mechanism question. Feedback mechanism. Feedback mechanisms are really important to a physiology class. All right, which of the following scenarios best describes a positive feedback mechanism? So I know from my notes that a positive feedback mechanism is a mechanism where the product causes the process to keep going. Right, the product of a process keeps that process going, so it creates more product, which creates more process, which is like keeps feeding on itself. So let's go find ABCD. Several hours after a meal, blood glucose levels begin to drop, prompting the pancreas to secrete glucagon. Glucagon causes the liver to convert stored glycogen to glucose, raising blood glucose levels. And so let's look at this one. In this one, the stimulus is blood glucose dropping. And so the process is to release glucagon, right? What is the product of releasing glucagon? Blood sugar increases, And so since it increases, that is going to go back and turn off the secretion of glucagon. And so A would be an example of a negative feedback, because this is when the product of a process turns off the process, right? B, after childbirth, high levels of prolactin induce lactation. So this is just highlighting the fact that when I give you guys examples, these are probably the examples that you're going to see in test questions, because I literally went over every one of these. After childbirth, high levels of prolactin induce lactation in the mother. During nursing, infant suckling triggers the release of more prolactin, which further stimulates lactation. This is where the product of the process is producing prolactin. The product of that is causing the infant to eat more, and it's going to cause more prolactin, which is going to stimulate more lactation, which is going to cause the infant to eat more. This is a process that is tumbling on itself, right? And so B is the correct answer. That is a positive feedback mechanism. I'm happy to go into more detail on those, but I think that question really helped to sort of define those. And let's look at B and or C and D as well. So during exercise, an increase of body temperature causes the body to produce sweat. Stimulus, increased temperature. The process is producing sweat, right? What is the product of that process? Causes the body to cool down. And so the body cooling down then is going to cause the process of producing sweat to stop. That'd be a negative feedback. Let's look at D. After eating, blood glucose levels rise, stimulating the release of insulin by the pancreas. Insulin causes cells to uptake glucose, lowering blood glucose. And so the process, or the stimulus here, is blood glucose goes up. What is that process? That insulin is produced. What is the product of insulin being produced? That cells take up glucose out of the blood. What is the what happens as a result of that? The blood glucose is lowered, which is going to turn off the process of releasing insulin. All of those things. So if you say to yourself, was something turned off? Did the product turn something off? That's going to be negative. If it turned it on, or if it caused it to happen more, then it's going to be a positive feedback. Let's continue. I'm happy to go over that more if you need me to do that more. Next question. All right. So here we have some ligands, we have some receptors, we have a couple things here. These arrows means that something's being activated, right? And this means a what's the word? Inhibition, stopping something, right? So looks like ligand 1 causes the signal transduction pathway, which increases CDK activity. Ligand 2 stops the signal transduction pathway. So then increased CDK activity would not occur. So ligand 2 and ligand 1, or ligand 1 and ligand 2 have opposite effects of one another. Let's go see what the question says. Diagram above represents a model of signal transduction pathway in the cell that is targeted by two different ligands, L1, L2. When L1 binds to the membrane receptor of R1, the pathway is activated and CDK activity in the cell increases. Literally, it tells me. But you know what? I like looking at those drawings and being able to decipher them by myself. Alternatively, when L2 binds the membrane, R2 downstream inhibited. Based on the model above, what would most likely result if mutation prevents L2 from binding with R2? Then CDK activity would never stop, and cyclic dependent kinase activity is as long as there's cyclin, the cell is going to continue to replicate. And so let's go find that. Signal transduction pathway would be inhibited, resulting in increased No. That one's got me turned around. So if this is mutated, then this is not happening. No, okay. Signal transduction pathway would proceed unchecked, resulting in decreased. No. Signal transduction pathway would proceed unchecked, resulting in increased cell division. C is the correct answer. Let's go C. Hey, I can actually like zoom in on one at like a proper font instead of like minuscule. This one's gonna seem like in a, look how much bigger that one is. I have to be careful because I like my books large print. Okay. In response to critically low nutrient levels, yeast cells often enter into a dormant, non-dividing state, G0. This state allows yeast cells to survive until nutrient levels are restored, at which point the cell typically re-enters into the cell cycle. It's just literally a definition of G0, right? But in this case, it gave us the reason that a cell would go into G0, which is low nutrient content. And because it wasn't hitting its checkpoints, right? That cell wasn't hitting its checkpoints, and so it went into G0. According to the information above, critically low nutrient levels cause yeast cells to transition from... Oh my gosh, why they put meiosis in here? Literally, okay, I'm like, didn't the prompt just tell us that? But I added G0 to this, and so you have to know the definition of G0 here. A is the correct answer. It's a good question. Good question. I achieved a streak apparently. I'm on fire. All right. Kenny, that answer your question? Let me know if that didn't answer your question, and I can go over signal transduction or feedback more if you need me to, or if anybody else needs me to. Glad to do that. All right, we're going to look at the unit test now. I feel like this material is pretty easy. As long as you read carefully. There's a lot of logic to this particular section. Just get all these things all stacked in here right. Mutations within genes coding for G protein coupled receptors, or GPCRs for those people who know them. Yes, I will definitely go over cyclins and CDK. I'm glad you mentioned that because I haven't seen any questions over that, and I will spend some time on that. I will actually stop the quiz for that. I think that would be appropriate. Can lead to one or two major outcomes. So mutation in those G protein, loss of function, yes, I can do that too. Loss of function or a gain of function. Loss of function mutations prevent signaling pathways, even in the presence of signaling molecules. Gain of function mutations activate signaling pathways, even in the absence of signaling molecules. Okay. Interesting. Let's see where they're going with this one. Which of the following is most likely result from a loss of function mutation? So if something loses function, then there's basically there's going to be no cellular response, right? There's going to be no transduction. There's going to be no response because of that loss of function. Signal transduction pathway that are inactive even if ligand to A. I don't. Maybe I'm missing something on this one. No. Okay. So let's pause our quiz for a moment here and let's look at these questions. Okay. Cyclins and CDK. So cyclins are proteins that are involved in the cell cycle. Cyclins are groups of proteins, right? It's not just cyclin, but there's lots of different cyclins, but they generally all do the same thing. CDK is an enzyme that's always in the cell. It's called cyclin-dependent kinase. When cyclin and CDK join together, they become a maturation promotion factor. And maturation promotion factor is going to promote maturation, right? Which in the context of this section is that maturation means cell division. And so the more MPF you have, the more closely your cell is moving toward cell division, right? And eventually, cyclin is going to degrade at a particular time in the cell cycle, whereas CDK is going to stick around. And so you see that in this particular graph. What this graph represents is 3 complete cell cycles, G1S, G2M, right? Then the same thing all over again. CDK levels remain the same throughout, right? Because it's an enzyme, it doesn't go away after it does its job. Notice after the DNA is replicated, or during DNA replication, cyclin production begins. As cyclin production begins, you'd expect it to begin binding with CDK to form MPF. So as MPF, it's going to increase in the cell. This is going to cause the cell to move toward mitosis and move toward cell division, which is exactly what it does until it reaches a point. That point is going to be anaphase, right? As soon as those cells start separating, or as soon as the chromosomes start separating, the cells start sort of becoming 2 separate cells. Cyclin levels in the cell are going to decrease because maturation no longer needs to happen. In fact, we need to undo maturation. And so cyclin's going to decrease. CDK is going to stay the same because the enzyme it always does. So cyclin and MPF are going to go down at the same time because they're related to each other, right? And then the cell continues on, G1S, G2, same sort of cycle. That was fast, but in general, that's what's going on. As long as you understand that cyclin is directly related to promoting the cell cycle, and then as long as it's present in the cell will begin doing the cell cycle. But it's supposed to go away as a part of the cell cycle. Go over there to look at cellular processes models and being able, yeah, we can do that. Let's see here. So secondary messengers, this picture's not a very good picture. This one works though. And again, this is the caffeine pathway, right? And you can see cyclic AMP in here. You're not going to be asked to specifically point out like where's the secondary messenger, you know. You might be asked to talk about its function. Like what is the function of cyclic AMP in this particular case? Well, in this particular case, it activates protein kinase A. It's activated by adenylyl cyclase, which you've probably seen adenylyl cyclase several times by now as well. Yeah. Can there be multiple secondary messengers? Sure. There could be. Usually it's going to be similar kinds of things. Let's see if we can find one here. Yeah. In this case, like IP3 is an example of one. Right? It goes and does a thing. And so it's just generally like a protein that, or not a protein, but a molecule that like serves as a relay point. It's not really doing the activation. It's not doing the receiving. It's usually not on the membrane as well. So like ligand moves over and turns on phospholipase. Phospholipase wouldn't be considered a secondary messenger. But again, you're not going to get a question that says which one of these is the secondary messenger. Right. Now, you might be asked what is the function of IP3, IP3 is a secondary messenger. What is its function in this particular process? It binds to calcium channels, which causes calcium to be released, right? And so that's the kind of question that you're going to see associated with them. G protein is, let's see if I can, here's a G protein. G protein is like a little relay here on the membrane. You wouldn't really call it a secondary messenger because it's on the membrane, but it's its own thing. It's anytime you see that, notice it is going between this receptor and this enzyme, right? It's just another go-between. That's all you need to know about it. And remember, you guys don't lose sight of this. You're not answering trivia questions, right? You're answering questions about a specific prompt. And so, what is the function of G protein in this particular case? Well, G protein is activated by the G protein coupled receptor, which then goes and activates this enzyme. If a mutation in the G protein occurred, what would happen? Then the enzyme would not be activated, right? Be the most immediate thing. And notice here, enzyme activates secondary messenger, which initiates secondary cellular response. You're always looking at the prompt, the specific prompt. You don't have to memorize molecules. That would not be a helpful exercise. I hope that helps. Let me know if it doesn't, or if you have further questions about it. Let's go look at the next question. This one's a lengthy one, or verbose. Many species of bacteria regulate gene expression using cell signaling system known as the quorum sensing. Sounds official. In this system, perfect example here, right? Gives you a new vocab word, then tells you what it is. In this system, bacteria synthesize quorum sense signaling molecules and release them into the surrounding environment. This affects these molecules. The effects of these molecules on gene expression depend on density of bacterial population, which is quorum. It's the idea of a quorum is having enough, like having enough individuals. So, use a cell signaling system known as quorum sensing. Those things gene expression only occurs if there's enough bacteria present, is what it's saying. Which of the following best describes what occurs as density of quorum sensing bacteria population increases? As the density increases, you'd expect gene expression to increase. Concentration of quorum signaling signal molecules decreases eventual triggering of change. No. Concentration of QS signaling molecule increases eventual triggering and change in the expression of few members. No. So again, they're all detecting this, right? And so the idea of few is just, and a few members is not good. The concentration increases eventual triggering gene expression throughout the bacterial population. Yep, I like C. It's kind of a weird question, but it's just a matter of, but you haven't figured that out yet. It's a lot of just chasing, everything's the same except for these words, or these 4 words, and you had to figure out what makes them wrong. Here's a fun question. Bunch of pictures. Thanks, Brian M. Swice, for your picture. So let's see what's going on here. Got the hypothalamus. It talks to the anterior pituitary, which talks to the adrenal cortex, which produces cortisol, which is going to go back when high cortisol levels are present. It's going to go back and turn off the hypothalamus and anterior pituitary, which is going to shut down the production of cortisol. This is a negative feedback loop. Let's go read the question now that we understand what we're looking at. In response to stress, the hypothalamic pituitary adrenal axis, HPA for short, coordinates the release of steroid hormone cortisol, as summarized in the figure above. Inhaled corticosteroids are a class of synthetic steroid hormones that are commonly used as long-term treatment of persistent asthma. Assuming the ICS function the same way as naturally occurring cortisol, it is most likely that long-term high-dose ICS would reduce cortisol production. Right? And this, I mean, I'm going to make sure I get it right first. Okay. Yeah. So this is literally one that each one of these is not like the other. You know, you've heard me say that before. Like, all three of these are the same thing, right? If you stimulate CRH, you're stimulating this. All of those are leading the same thing. And so reduce is the only right one. All right then. Moving on. Student is examining a whitefish blastula cell under the microscope. Based on his observation, student proposed that the whitefish blastula cell is in interphase. Which of the following pieces of evidence best support? If it's in interphase, you don't see any chromosomes. You see a true nucleus in the center, right? Let's go find that. Cleavage furrows formed. Nope. Mitotic spindle. Nope. Sister chromatids have separated. They haven't. A, B, and C are wrong. D is the correct answer. We don't even have to read it, but let's read it. Genetic content of the cell is dispersed as chromatin. D is the correct answer. Good, good, good. Here's a nice picture. Kind of running out of space. A nice picture. You got a presynaptic cell. Again, this stuff is unimportant. What do you see here? You see some sort of molecule. It's being released. It's being received. It's I think it's Wikimedia. Never heard of that. Which the following best describes cell communication occurring in the figure above. There's several things we could see, right? This could be like local, regional sort of communication. Let's see. The signal emitting cell communicates with a far away cell by releasing long. I just don't like this because it's not defining those terms. It's not long distance here. Yeah, I don't like it. Signal emitting cell communicates with a nearby cell by releasing local regulators. Yeah, B is probably correct. Let's look at the C and D. Far away, direct cell-to-cell contact. So C is wrong because it's far away, and D is suggesting that it's direct cell-to-cell contact. They are not direct contact, and so B is the correct answer. We haven't used the term local regulators, but I'm sure you can figure that out using your context clues. It's a local regulator. See if I can zoom in a little bit. I like that these questions are exposing you to things that you probably haven't seen, because it's shows you that you can answer questions about things you don't know about just by knowing a few things. Adenoviruses are a family of highly contagious viruses that most commonly cause illnesses of the respiratory system, such as bronchitis. Anybody got time for that? Or pneumonia. In the first phase of adenovirus infection, the host cell expresses several viral proteins. One of the proteins is that one, which inhibits the activation of the host protein p53, a tumor suppressor that arrests cell cycle and induces apoptosis when DNA is detected. Okay, so what's going on here? When this virus infects a cell, one of the proteins that is expressed inhibits p53. p53 is a tumor suppressor that stops the cell cycle and induces apoptosis. And so if you are infected with this virus, you are not able to do tumor suppressing, right? And you're not able to arrest the cell cycle. And so you're not able to induce apoptosis. And so if p53 doesn't work, the cell cycle can't be regulated properly, right? If there's DNA damage, the cell can't pull itself out of the cell cycle or go through apoptosis. And so let's go and find that. The E1B causes infected cell to undergo programmed cell death. No, it's the opposite. E1B increases the likelihood of cell cycle arrest. No, it doesn't. E1B decreases the likelihood of apoptosis in infected cells. C is the correct answer. Let's look at D, though. Let's see why D is also wrong. E1B causes infected cell to transition out of the cell cycle and go into G0. Again, that'd be a helpful cell thing. E1B causes the cell to hurt itself, essentially, so C is the correct answer. And this is another one. Which one of these is not like the other? Ooh, I love this graph. I'm so thankful that we got to see it, because you guys need to see this graph. This is another common one that I didn't have a picture of, and so I'm so thankful that we get to see it. The following graph shows how cellular DNA changes over the course of typical mitotic cell and rapidly proliferating human cells. Proliferating is just dividing, increasing, right, becoming more. And so let's look at this. You have time on the X, you have DNA content in picograms. And so during G1, your DNA content is 6, right? And what I have seen this drawing where G1S, G2M is not on here, and you were having to, it was like 1, 2, 3, 4, and you were having to guess what is cell or what is part 2, based on what you see. And you'd be able to figure that out, because what's happening in S phase, DNA content is doubling, right? What's happening at the end of M, DNA content is cutting in half again, and it's sharp. It's like whoosh, because what is happening here? This is cytokinesis right here, right? And so I love this picture. And I'm not saying you'll see it on the test tomorrow, because I haven't looked at the test in a year, but I have seen this test a few times. I know an older test that I used to give had this picture on it, but I don't think the one I give now does. But I think it's a really important picture, because again, it's just a graph. Can you read a graph? Can you look at this and not be wigged out by it? Based on the graph, which the following statement is correct. So let's go see. Human cells spend the majority of the cell cycle in interphase. That is definitely true, right? It spends 23 minutes in interphase and a very small portion of its life in M. A is correct. I don't know what the other ones say, but let's look and see why they're wrong. Human cells spend the same amount of time of in phase as they do interphase. No. And so how could you get this one wrong? You don't know that G1S, G2 is all interphase, right? The amount, which I take that for granted, but some of you guys don't know that. The amount of cellular DNA remains constant throughout the cell cycle. No, it doesn't. The amount of cellular DNA is half during interphase. It's not. It increases by twice. A is the correct answer. Love that graph. A few more questions here. Okay, so this is a feedback question. Which the following correctly describes how positive and negative feedback are different? Negative feedback turns off something. Positive feedback causes it to continue to happen. Those are the big differences, right? Negative feedback reverses changes in a system. Amplifies. I don't like the word reverses there, so we're going to hang on to A, because reverses would mean it undoes everything, and that's amplifies is a fine word. I don't like reverses, but I'm not. Sometimes word choice is hard, and so we're going to go see if it was too hard for the writer of this question. Let's see. Positive feedback occurs only in response to stimuli, while negative feedback can occur in the absence of stimuli. Don't like that one. Negative feedback requires the detection of a stimuli. Positive feedback moves a system closer to its set point, while negative feedback moves it further from the set point. I knew. This question is full of weird language, but yeah, so I can see where, and so here's their reasoning for use as the word reverse, and let's look at this. Negative feedback reverses changes in a system, helping to maintain the system at its set point. And okay, the change. Let's do an insulin one. So it's simple. The change in the system is that blood glucose levels have increased, and so it's reversing that change. If they're talking about the stimulus as being the change, that's fine. I'm okay with that. But it was unclear because you could have read that and thought the change was the mechanism itself. So in that context, it's fine. The other three were really bad, and so A is the correct answer. Ooh, this is going to win the prize, I think, for the biggest question of the night. I'm going to try to get it all in there and then still be able to read it. I love that they're pulling excerpts from like scientific articles and stuff. It's really good. Okay. The following excerpt and image come from a scientific paper written by that guy and Chess. They discuss the role of G proteins in olfaction, or the sense of smell. Each mammalian olfactory neuron appears to use the same machinery for transducing signals from its odorant receptor molecules. Big words, but we're not afraid of these words. Upon odorant binding, the receptor is thought to activate GALF, a G protein. GALF mediated activation of adenylate cyclase 3 then raises intracellular CAMP levels, causing a cyclic nucleotide gated channel to open. The influx of cations through this channel ultimately leads to the formation of an action potential, which allows the primary neuron to signal to the brain. Okay, so let's go see this on this picture. All right, here we go. Love this picture. This is great. So, all right, this is a neuron, right? And now we're zooming in on that little spot. And we see a thing. Here's our odorant. This is something that they smell. There's the receptor. There's GALF. GALF is that G protein, right? When it's activated, it's going to go turn on adenylate cyclase, which is then going to turn on cyclic AMP, which is a secondary messenger here. And that secondary messenger is going to come over here and open up this channel, which is going to cause these ions to come in, which is going to cause an action potential. And it defined action potential here for you, which allows the primary neuron to signal to the brain. So action potential, again, it defines it here for you, so I'm not going to go into a lot of detail, but it's just sending that signal to the brain. It's basically the signal that's received here. It's going to just go whoosh, and it's going to go to the brain. Projects to the olfactory bulb, which is part of the brain. Mutation in a component of this signal transduction pathway prevents the cyclic nucleotide gated channel from opening. if this channel doesn't open, these ions don't come in, action potential doesn't occur, that signal never gets to the brain. So reception still happens, but the brain doesn't know that it happened. Let's go find that. Wait, first of all, which of the following scientific questions would best help researchers to understand how the mutation prevents the from opening? Let's understand what's happening here first, because this is normal. You'll probably see 3 of these on the exam in May. Which scientific question, right? Would best help researchers understand how the mutation prevents. The mutation causes the shape of this to change, which is going to cause it to not work. That's all we're looking for. Does the mutation stimulate the conversion of ATP to... No, that, again, not important. Does the mutation block the activation that's on back here, so that wouldn't happen anything? Does mutation promote the influx of cations in the cell? That's what's actually happening. And so we like C. Does the mutation cause intercellular camp levels to rise? I think C is... C is the correct answer, because it's the only thing that has anything to do with the question at hand. But C was not the correct answer. So let's see. Which of the following scientific... Okay, so I'm reading this wrong. I would have missed this one on test. That mutation prevents it from opening, and so let me go back up here and show you the picture again. And so it could be anything back here, right, that could prevent it from opening. And so let's look at that question with that understanding. I think that'll help. Stimulate the conversion? No, it doesn't. Does it block the activation of... I think this is what we're looking at. And this is the classic, which one of these is not like the other that I missed this time. Probably some of you would have got it right. Because A, C, and D all have to do with it working properly. B does not have to do with it working properly. Watch me miss it again. No, I just didn't read that one carefully enough. Yeah. It's funny, I watched this, maybe it was TikTok, I don't know. And it was this teacher talking about things that schools need. Or no, things that schools don't need or something. It was something like that. And one of her things was teachers that couldn't pass the tests that they give to their students. And I thought, whoops, that's a thing. That is a thing. I've had people ask for content stuff before and then ask me for keys later because they couldn't answer it themselves. During the humoral immune response, helper T cells activate B cells through a cell communication. This process is shown below. So you have helper T cells here. Okay, and they're touching each other. Introduce a substance that causes the T cell receptor to break down. Okay, so the T cell receptor is not going to be there. The cells aren't going to be able to touch anymore. Which of the following best describes how a humoral immune response will be affected? Let's go look. This picture seems backwards to me, but let's go see. Helper T cell will no longer bind to the B cell. As a result, the B cell will not be activated. I like A. Let's look at the other ones. Helper C cell will no longer bind to the B cell, and B cell will instead be activated by helper T cells, releasing long distance signals. I don't think so. Helper T cell will still bind to the B cell. No. I think it's A. Made a 90. 9 out of 10. Next up, Unit 5. Not yet. We have to do Unit 4. Hey, guess what? It's been about an hour. I don't know if any of you are still around. But it's pretty much time for me to sign off. If you have questions about the content, please come see me in the morning. I feel pretty confident just going back through this. I feel like I've covered it pretty well. I feel like if you missed some stuff, hopefully you've watched the videos by now. If not, it's a little late. But you know what? There's still time before tomorrow morning. Please come by and see me if you have questions at all, or send me an email. I'll get it first thing. I can answer it. Not going to answer it tonight because I'm not checking my email tonight, but I will get it when I show up at school tomorrow early, and so you can look at it if you don't have a chance to come by and talk to me. Or come by, we'll talk about Unit 4. It's been fun. Thanks for watching. Later.